The bacteria that cause tuberculosis, Mycobacterium tuberculosis, primarily affect the lungs through a complex interaction with the body's immune system, which can result in either a contained infection or active disease. Here is a breakdown of the process in the lungs: inhalation and initial infection. The bacteria are spread through the air in tiny droplets when a person with active TB disease coughs, sneezes, or speaks. Once inhaled, the bacteria, called tubercle bacilli, travel into the lungs and settle in the alveoli, small air sacs. Alveolar macrophages, which are immune cells, attempt to engulf and destroy the bacteria. Bacterial evasion and multiplication. M. tuberculosis is highly skilled at surviving inside the macrophages. It prevents the normal killing mechanism, the fusion of the phagosome containing the bacteria with the lysosome containing digestive enzymes. Instead of being destroyed, the bacteria begin to multiply inside the macrophages. Granuloma formation, containment. The immune system responds by recruiting other immune cells, like T cells and more macrophages, to the site of infection. These cells aggregate and form a protective wall around the infected cells, creating a structure called a granuloma, sometimes referred to as a tubercle. The granuloma essentially traps and isolates the bacteria, preventing them from spreading. At this point, the person has latent TB infection. The bacteria are alive but are kept under control by the immune system. The person usually has no symptoms and cannot spread the disease. Progression to active TB disease, damage. If the immune system weakens, due to factors like HIV, malnutrition, age, or other health conditions, the granuloma can fail to contain the bacteria. The bacteria begin to multiply rapidly and break out of the granuloma. This is known as active TB disease. The bacterial multiplication and the body's immune response lead to significant tissue destruction and necrosis, cell death, in the lung. A hallmark of active pulmonary TB is caseous necrosis, a type of cheesy, destructive lung tissue, and the formation of cavities, gas-filled spaces in the lung parenchyma. When these cavities rupture into the airways, the bacteria are released into the sputum, making the person highly contagious. Long-term effects. The tissue damage can lead to complications such as fibrosis, scarring and thickening, stiffening of the lung walls, bronchiectasis, irreversible widening of the airways, and reduced lung function, which can persist even after the disease is cured with treatment.